Hello everyone, welcome to ITLS Academy. ITLS Academy is the best online training platform with unique features you will not find anywhere else. I am Siddharth Singh, pursuing PhD in Cancer Biology from Thapar University, Patiala. So today's topic will be epidemiology of human cancer. So let's start. Before going into the details of the epidemiology of cancer, let's try to understand what the term cancer and what is the term epidemiology basically mean. Cancer is basically a group of diseases which involves abnormal cell growth with the potential to invade or spread to other parts of the body. It means basically cancer is an abnormal cellular growth. In normal cases, all the cell divisions or the cell cycle is been tightly regulated by various checkpoints. So these checkpoints, they undergo certain mutation due to which they lost their functionality. After losing their functionality, they don't behave in a normal way. So what happens is the process which is previously tightly regulated has now become unregulated. So normal cells, they undergo abnormal cell division and a huge mass of cell is developed, which is not required by the body. So this is basically the main idea of cancer, how the layman terms of cancer is, which I have explained just now. So let's move on to the next definition is epidemiology. Epidemiology is the branch of medicine which deals with the incidence, distribution and possible control of disease and other factors relating to health. So basically what epidemiology means, it is the branch of medicine which deals with or which study the incidence. What does incidence means? Incidence means for a particular disease, what is the places where it is happening and what is the scenario in which it is happening. For distribution is basically how the disease which is occurring for a particular location, how it is spreading. Means either men is getting affected more or women is getting affected more or child are getting affected more. What is the age group which is getting more affected? Is there an relation to the old age? Is there any relation to the younger age generation people which is getting more affected by a particular disease? This comes under the distribution. Next is possible control. So what are the other methods which can be employed to control a particular disease from spreading? Another definition of epidemiology is it is a study of cancer in population and carcinogenesis, chemical and radiation carcinogen. So it is the study of cancer in population, like in India, if we do a particular study like how many percentage of people are getting affected from lung cancer below the particular age group, then it is a population based study. We are generating data to generate some results based on which we can on which the government can take decisions that which vaccine is to be given to at what particular time it is given. So and it includes also the carcinogenesis, the process of development of cancer, how the cancer is being generated in a human body, and the study of agents which are causing cancer like chemical carcinogen and radiation carcinogen. So basically these are the two types of carcinogens which play a major role in the development of cancer. There are other factors also which play the role in the development of cancer, like viruses and parasites and bacteria. But generally most of the cancers are related with the exposure of chemical and radiation carcinogen. Let's move further. So what do we understand by studying the epidemiology? First and foremost, we will try to understand, or after studying epidemiology, one will better understand the concept of how cancer develops and why it is different in different population. Because we are doing a population based study, so we will generate data that this particular type of lung cancer or this type of cancer is prevalent in particular country, and this type of cancer is prevalent in some other type of country. So we will understand what is the distribution of a particular disease or particular type of cancer for a particular country, and also what is the mechanism which cancer cells adopt to develop into a cancer disease. What else we can understand? we will understand what are the different environmental factors that are predisposed to cancer. So we all know that cancer is a group of disease, but other environmental factors play a crucial role in the development of cancer. The cancer genes are already present in all human beings, but all the human beings are not developing cancer because they are not exposed to particular stimulants or particular environmental factors that activate those cancer genes. So we will understand what is the role of those environmental factors in the development of cancer, or which are the environmental factors if a particular human being or particular individual is getting exposed to will have more chance of developing a cancer. So basically it gives a clue to the etiology of many cancer. What does etiology means? Etiology is the study of causation or origination. Basically how the cancer is getting developed or originating, and what are the other factors which help in the development of those cancer. So what is incidence? Incidence is like I told in the previous slide, how much this case is present in a particular country and how much it is present in some other country. So this comes under the heading of incidence. So basically cancer is higher in developed countries and it is low in the underdeveloped countries. Nowadays it is getting prevalent in in underdeveloped countries, but predominantly cancer is a disease of a developed world. Why this is happening, or why is such that only developed countries is having more of the cancer patient, is because of their dietary habits and their environmental factors and their sedentary lifestyle. These are the basic or the major factors which contribute to the prevalence of cancer in the developed countries. And in underdeveloped countries, basically people are doing more hard work and they are not so much exposed to the Western diet, so they are still underdeveloped countries are showing low rate of cancer. According to WHO data, in 2012, 14.1 million new cancer cases worldwide are reported, out of which 8.2 million people have died. So, according to WHO, the commonest lung cancer or the commonest cancer in men is lung, whereas for women it is breast cancer. Death rate is decreasing in developed countries. The rate of cancer development is higher in developed countries, but the rate of death is decreasing in developed countries. Why? Because they have more advanced medical facilities. They can detect cancer in the early stage. It has been well known that earlier the cancer has been detected, the more are the chances that a particular patient or particular individual can be saved. If a particular patient can be detected in a stage 1 or a stage 2, then the chances of saving the life of that person is drastically more as compared to if a person is getting diagnosed in the stage 3 and stage 4. Stage 4 is basically the metastatic stage, and in which the cancer has spread to various organs. Stage 1 is the stage in which there is just a small beginning of cancer, or only a particular part of a body is being developed a cancer. Some cancers like cervical and gastric cancer are decreasing in the developed world, obviously because of their better health facilities. Now, environmental factors. Environmental factors are the factors which play a role in the development of cancer. Basically, all the human beings, they have cancer causing genes, but all the human beings are not developing cancer. They are developing, only some individuals are developing cancer. So environmental factor play a crucial role in the development of cancer. Like suppose gastric cancer is much more higher in Japan than in any other country, and breast cancer is higher in USA. Why it is so? Because a particular individual is getting exposed to certain combination of environmental factors which supports gastric cancer in case of Japan and which supports breast cancer in case of USA. A major factor which plays a crucial role in the development of cancer is the diet. There are various researches in which obesity has been linked to development of various disorders, including cancer. So, an obese person is at a higher risk of developing cancer than a non-obese person. Smoking is another factor which is linked with developing of cancer. The person who is smoking, there are higher chances that it will develop, or he will develop upper aerodigestive cancer, or lung cancer, or urinary bladder cancer, or pancreas cancer. Why? Because smoking is itself is a carcinogen, a chemical carcinogen, which it is directly getting exposed to our human body. So our human body, they react to those carcinogen, and the human cells, they undergo some mutation, due to which they develop cancer. Tobacco is the another cause which is linked to the oral cavity cancer. Alcohol is also linked to cancer, but the majority of the cases are linked with diet and smoking. Alcohol is linked to very few of those cases. The people who are drinking alcohol has been reported to develop oropharynx cancer, larynx cancer, esophagus cancer, and liver cancer. Certain individuals with the reproductive history, like a few females which are having a continuous exposure of estrogen hormone, they are prone to develop breast cancer and endometrium cancer. Now, certain infectious agents like viruses, bacteria, and parasites has also been reported to contribute in the development of cancer. An example for virus is human papilloma virus. This virus is linked to head and neck cancer, for the development of head and neck cancer in humans. Certain bacteria is like Halobacterial pylori. It is the commonest form, or the form which is present in most human beings, but the continuous exposure of this pylori bacteria towards the human may result into the development of cancer. Now, age and cancer, how they are related. A particular individual which is in its younger age will have a particular or a more efficient immune system than an individual which is in the bracket of age 55 to 75. He will be generating better immune cells, or better B cells or T cells, to fight the mutation which is occurring towards in the body. But due to the age, the immune system starts deteriorating itself. The quality of B cells, or the amount of B cells and T cells which are being developed in response to a particular foreign infection, their quality is getting decreased as the age progresses. So the age is being linked to cancer. The higher the age, the higher is the chance of a particular person of developing cancer. Another relation is accumulation of somatic mutation. Now the process of cancer is basically related to the mutation of particular gene or particular allele. Now, try to understand this. If a particular mutation is happening over a particular location at, suppose, X, then overnight this mutation will not develop or not cause cancer in that particular individual. But over a time, like suppose for 10 years or for 20 years, the same mutation is going to happen at multiple places, or like suppose thousands of places, then this can result into the development of cancer, because now the quantity of mutation is sufficient to change the phenotypic or phenotypic results, or the gene product, or the protein which is being synthesized from a particular gene. This mutation is sufficient to change the structure of that protein, which ultimately results in the mutation. Decline in the immune function, this I have already discussed just above. Like with age, the immune system starts functioning, or the functioning of immune system starts getting deteriorating. Now, what are the acquired predisposition conditions? Acquired predisposition conditions basically means that there are certain things or certain genes which are already they have predisposed condition that they will develop into a particular type of cancer. Like chronic inflammation of mesotheliomas or lymphomas are the cancer which are being developed because of the long-term inflammation of a particular cell. Now, what is metheliomas? Metheliomas are the cancer or the tumor of tissue that lines the lung, stomach, heart, and other organs. So, if there is a chronic inflammation in these cells or these tissue, then they might develop mesotheliomas. Same-wise, lymphomas is the cancer or the tumor of lymph nodes. There are certain immunodeficiency states which contribute into the development of cancer, like immunocompromised person, like the person which is having HIV, AIDS. They are immunocompromised person. Their immune system is not functioning properly. So these are the states in which the immune system is, since the immune system is not functioning properly, so any foreign. Now another factor is precursor lesions, which can lead to development of squamous metaplasia and dysplasia of bronchial mucosa, endometrial hyperplasia and dysplasia. Moving further, what are the genetic factors which can be linked to development of cancer, or which are the genes which are responsible of development of cancer? We already know that a lot of research has been done on the genetic factors which can cause cancer. So the most common example is the BRCA gene or BRCA1 gene. It is a gene which is being linked to the development of breast cancer. Another gene is edematous polyposis coli or APC and familial retinoblastoma. Retinoblastoma is the cancer of eye. So there is a very complex relationship between genetic factors and environment. Only a particular combination will result into the development of cancer. Not all the combination will result in the development of cancer in a particular individual, but in most of the cases, when there is a mutation in BRCA gene or APC gene or RB1 gene, then it will result in the development of the concerned cancer. Now here are the list of some genes which can cause cancer. The P53, it is a tumor suppressor gene. Tumor suppressor gene basically means that the role of this gene is to suppress the tumors which are normally occurring in human body. Once this gene loses its functionality, then the tumor suppressor function of this gene will not happen. Hence, the gene or the mutation which is or the genes or which is related or the cancer which is being developed in human body naturally will not be checked or will not be controlled. So it will develop at its full flow. This one is RB retinoblastoma, APC is adenomatosis polyposis coli, P10 it relates to the mutation in P10 gene results in the development of epithelial cancer. P10 is basically phosphate and tensin homologue protein. Now, the genetic and molecular epidemiology of cancer. What do we mean by genetic and molecular epidemiology of cancer? It basically means the sequence of events or the events which are being happening or which are occurring at the molecular level. So at the molecular level or at the gene level, the most common event or mutation which occurs in human genes or in an individual is polymorphism. Polymorphism is basically alternative form of a gene. Like basically, if a gene is present in a particular situation in a particular individual, the sequence of a gene is suppose ATGC. Now, if a particular mutation is happening and T is being mutated to A, now the sequence become AAGC. So this is known as the polymorphism of this particular gene. The best common or the normal example of a particular to study polymorphism is MMR. MMR is the mismatch based repair mechanisms which are put in human body to check if there is a mutation in a particular gene, then these pathways play a role to correct those mutations and the normal life goes on. Inside these MMR pathways, there are two common examples, MLH1 gene and MSH2 gene. They play a role in the development of cancer when they are present in a mutated form. In normal form, MLH1 plays a role in the development of a protein MLH1 and MSH2 play in the role in the development of a protein MSH2, which play their overall role in the stopping of cancer. Now, if a polymorphism is present in any of the gene, then that particular individual will develop cancer. Now, moving on to the carcinogenic agents. What are carcinogenic agents? Carcinogenic agents they inflict genetic changes or genetic damages in the cell. Basically, these are the agents or these are the environmental exposure factors which can cause cancer in a particular human individual. There are basically three types of carcinogenic agents. One is chemical, another one is radiation, another one is microbes. So chemical radiations or chemical agents are all the chemicals which we are getting continuously exposed. The most common or the most remarkable example of a chemical carcinogen is the asbestos. So asbestos is a chemical which is continuously being used or continuously being released in mines. So Dr. Parvikal Pot is the first one to understand or to report that asbestos is linked to a particular type of cancer, which is scrotal cancer. So most striking example of a scrotal cancer occurring in chimney sweeps, it is identified more than 200 years ago by Dr. Sir Parvikal Pot. So asbestos has been linked to development of scrotal cancer. This is an example of chemical carcinogen. Now these are the certain or we can better understand the chemical carcinogen from this slide. There are certain, basically the chemical carcinogen has been divided into two parts. First is direct acting or indirect acting. What do you mean by direct acting? Direct acting, basically once a particular human is being exposed to an agent, and if it is a direct acting agent, then it will not need any further processing steps. As soon as it enters into the human body, it will start affecting its function or start causing mutation in the human body. So these are known as direct acting. An example of direct acting chemical carcinogen is alkylating agents which are used in the treatment of cancer. This is ironic because the same disease which we are in which they are being used to treat, they also causes the same disease at the later stage of a particular individual or the later life stages of a particular individual. What are indirectly acting chemical carcinogen? They require a metabolic conversion to an ultimate carcinogen. Basically, they means if they enter into a human body, then they will get converted into their active form. That active form is basically a carcinogen which can cause mutation in a particular gene or particular protein of humans, which will ultimately lead into the cancer. The most potent is polycyclic hydrocarbons. Some examples of chemical carcinogen are benzopyrene in lung cancer, or aromatic amines, or conversion by P450 dependent monooxygenase, and aflatoxin B1, which is linked with the development of hepatocellular cancer. Now this is the list of chemical carcinogen this is a very short list. There are hundreds of other chemical carcinogen which are linked for the development of a particular cancer in humans. So first one is acetaldehyde, then acid mist, aflatoxin, alcoholic beverages, aluminum production, butadiene, aristocolic acid, arsenic acid, asbestos, cadmium, azathioprine, benzene, benzodiene. So these are basically the list of chemical carcinogen which play a role in the development of cancer directly. Going further, what is the mechanism of action for the development of cancer? So basically, let's look at this diagram. In this diagram, we will start from a chemical carcinogen or any carcinogen, for example. Be it a chemical carcinogen or an indirect carcinogen, whatever carcinogen it, we will start from a carcinogen. So let's suppose if we are starting with a indirect carcinogen, then it will get activated. The metabolic activation it occurs after entering the human body and will generate electrophilic intermediates. Now in human body, there is a normal process which keeps a check on these development of carcinogenic substances or entry of these carcinogenic substances. So if that process is active in a human body, then the detoxification of these intermediates will occur, and the excretion of these substances will occur through urine or through the rectum section of human body. Now if this detoxification process is absent or mutated, now what will happen? Now these electrophilic intermediates will bind to the DNA and will form adducts, or basically in layman term they will mutate the DNA. Now if once the DNA is mutated, the human body is also having a process which can repair itself or which can repair the DNA. Now if that process is present, then the DNA repair will occur and the cell will develop or return into its normal position. If this DNA repair process is absent, then there are two positions. There are two options. Either the cell can die through the process of apoptosis, or the permanent DNA lesions will occur and the process of the development of cancer will occur. So this phase is the initiation phase. Till here is the initiation phase. So this phase is the initiation phase. Now once the cell gets initiated, the initial process of initial processing of a chemical carcinogen or the cell is done. Now the further steps what it can do is its cell proliferation. Now what cell proliferation is? The cell will multiply or cell will proliferate into different kinds of cell having the defective genomic combination or having the defective gene. So multiple copies or multiple daughter cells from that initial cell will develop. Now the pre-neoplastic clones will develop, which another can proliferate, and additional mutation can occur due to the continuous exposure of a chemical carcinogen or any other carcinogen, which will lead into the development of malignant neoplasm. So this step is known as promotion. So mutagenic substances contain highly reactive electrophile group that form DNA adducts, which leads to the first process of initiation. Subsequent expansion of the mutated cells may occur due to the promoters. So this is what is the mechanism of development of cancer. Once the human body is being exposed to a particular carcinogen, be it a chemical carcinogen or a radiation carcinogen. Moving further, the radiation carcinogenesis. It is the best example of radiation carcinogenesis is Hiroshima and Nagasaki and Chernobyl. Chernobyl, we all know the nuclear power plant that failed in that Chernobyl area, and Hiroshima and Nagasaki, we already know that they have seen the bomb blast of atom bombs in those countries. So people till now being developing cancer because of the radiation present in these areas after the exposure of those events. Chernobyl, a nuclear power reactor failed, so all the radiation is still present in the soil and the air and the sand. So people are still developing, or people are prone to develop cancer or living in the nearby areas of Chernobyl. No one is living in the area of Chernobyl because it is contaminated, but still people are living in the nearby areas. So even in the nearby areas, people are developing cancer. Now therapeutic radiations can also cause cancer. The radiation like X-rays radiation or the radiation which the human beings are using for the treatment of cancer, like the radiation therapy, they can also lead to the development of cancer. It is still ironic that the same thing which we are using to treat a particular disease can cause a disease at the later life stage of a particular individual. Now what is the mechanism of radiation carcinogenesis, or how the radiation can cause cancer in a particular individual? It double-stranded. It breaks the double-stranded DNA and it causes translocation and mutation in the cancer gene. Like we all know that the DNA is double-stranded and they are having hydrogen bonding in between A to GC. Now G and C are linked with triple hydrogen bond and A and T are linked with double hydrogen bond. So these hydrogen bonds or the bonds between different bases, they gets broken after the exposure to a particular radiation. Another method is translocation. Translocation is the process in which a particular nucleotide or a particular base gets translocated from its normal position to some other position, which will lead to development of mutation, which will ultimately lead into the development of carcinogenesis. Now how UV rays can cause cancer? UV rays in the sunlight can interfere in the DNA by forming pyrimidine dimers within the DNA. Now we all know that the DNA is composed of two types of bases, purine and pyrimidine. The UV rays from sunlight can interfere or can mutate or can initiate the binding of pyrimidine dimer, due to which the whole structure of DNA will change, which will ultimately lead into development of a particular mutation. Due to that mutation, a cancer can develop in a particular individual. Now the most prevalent cancer which is relevant or which is caused by UV rays is skin cancer. So that's mark the end of today's lecture. I hope you like this lecture. Thank you.